Okay, ladies and gentlemen, thank you very much. It's very nice to be here in Milan. I'm going to tell you the story of Bob Smith. Bob Smith was a colleague, a clinical pharmacologist, at that time working at St. Mary's Hospital in London, and he performed a pharmacological experiment. He took the drug, the antihypertensive drug debrisokine in 1975. He took 32 milligrams. Also his colleagues at the lab, 19 other researchers, took this drug, 32 milligrams of debrisokine. And the experiment went very well, except for Bob, because Bob got dizzy. He had got orthostatic hypotension, and he fainted. It was a pharmacological experiment, and he studied the pharmacology of this antihypertensive drug. And the question, of course, was what was the explanation for this extreme drug response in Bob, and as compared to the other 19 individuals. And they were happy to collect urine samples from all participants. And in the most participants, they could measure the drug, debrisokine, and also the inactive metabolite for hydroxydebrisokine in the urine of the patient. But that was not true for Bob, because in Bob's urine, they could only measure debrisokine, and no 4-hydroxydebrisokine could be measured, meaning that Bob's liver was unable to metabolize this drug. And that, in fact, was the reason why this 33 milligrams of debrisokine was, in fact, too high a dose for Bob. And later they found that this inability of the liver of Bob to metabolize this drug was related to a genetic polymorphism in the gene encoding and a very important liver enzyme, CYP2D6, cytochrome P450-2D6, which is a very important enzyme in the metabolism of drugs. In fact, about 25% of the commonly prescribed drugs are metabolized by this enzyme. And that explained why Bob Smith was so sensitive for this drug. In fact, we know in pharmacology already for more than, let's say, 100 years that the drug response is highly variable between patients. And we always thought that characteristics such as shown on this slide, like age, body composition, comorbidity, ethnicity, but also environmental factors. Alcohol, food is of influence on how an individual responds to a drug. But now we know since, let's say, 40 years that also genetics plays a very important role in variability of drug response. It's an important source of variability in drug response. And not only like in the example of Bob in drug metabolism, but also with regard to absorption of drugs, excretion of drugs, all the genes encoding for these enzymes, these drug transporters, are highly polymorphic and may introduce variability in drug response. And not only in the pharmacokinetics of the drugs, but also in the pharmacodynamics, the mechanism of action. We now know that genes encoding drug targets are highly polymorphic and may influence drug response in individuals. And also in signal translation pathways, the genes encoding these enzymes and these proteins are highly polymorphic. And altogether, this introduces variability in drug response. So in case of Bob Smith, the CYP2D6 enzyme, currently we know that there are more than 100 genetic variants in the gene encoding this liver enzyme. And we still, in pharmacogenetics, we still use the star allele nomenclature. Star 1 is the wild type. And for example, star 4 is a SNP, a single nucleotide polymorphism, relatively frequent in Caucasians, up to 4%, and leading to an absence of functional enzyme of CYP2D6. From this slide, we can also see that there is large inter-ethnic variability because the star 4 allele is very rare in Asians, but in Asians, much more frequent is the star 10 allele, also leading to reduced enzyme activity. So we can genotype the CYP2D6 gene, for example, using AmpliChip or other methods, and Bob was a star4, had a star4 genotype, leading to an absence of CYP2D6 activity, meaning that he had a poor metabolizer phenotype. So nowadays we could have genotyped Bob Smith before giving the drug, collect saliva samples, DNA samples, extract the DNA, perform genotyping, and then we would have shown that Bob was a poor metabolizer, and we would have decreased the dose, not giving him 32 milligrams of CYP2D6, but probably 5 or even less milligrams of this drug. So this was a story of almost 40 years old, and we have done a recent survey among physicians and pharmacists asking, do you believe in the concept of pharmacogenetics? Meaning, do you believe that genetic variation may influence drug response? And almost all physicians and pharmacists answered yes. We believe in the concept of pharmacogenetics. Genetics is important with regard to drug response. However, if we ask them, did you order or recommend a pharmacogenetic test to treat your patients, then almost only 4% of the general practitioners did so. Pharmacists did a little better, 15% recommended or ordered a test. And that may look strange because they believe in the concept, but they do not use this knowledge in treating their patients. And in fact, in that survey, it was shown that the physicians and pharmacists, they have a very high belief in pharmacogenetics, high expectations, because they think that using pharmacogenetics will prevent choosing a wrong drug or choosing a wrong drug dose, and will lead to a more higher efficacy of pharmacotherapy, and will decrease adverse drug reactions. So mostly one of the first questions is there enough evidence, clinical evidence, that the use of pharmacogenetic screening is indeed in the benefit of our patients? There are at least 6 randomized clinical trials summarized on this slide, and I have the color green means that the study was positive, and red is what was negative, or at least there was no benefit. So there are examples, several examples showing that if you test patients before prescribing drugs, and you use this information to guide your prescription, adjust the dose, that this is in the benefit of our patients. We contributed to this field by doing randomized clinical trials in inflammatory bowel disease. Patients are treated with 6-mercaptopurine or azathioprine, and we randomized patients for screening versus no screening. Patients were tested for the TPMT gene. 3 genetic variants were tested. And if patients were carrier of one of these genetic variants, we reduced the dose to 50% in heterozygous patients, or 90% in case of an homozygous patients. The primary endpoint was leukopenia, and in this study, which was published in Gastroenterology in December 2015, we showed that there was a 10-fold decrease in hematological adverse drug reaction in leukopenic events if you screen patients and adjust the dose of 6-mercaptopurine and azathioprine. And the editor of the journal was kind enough to write an editorial saying that the time for debate is over, start testing TPMT. However, if we ask our clinicians in the hospitals in the Netherlands, do you test routinely for TPMT, only 10 or 15% of the physicians use this test before they prescribe the drug. My question always is, what would you need for what kind of evidence do you need more to be convinced and start implementing this test? So a difficulty in this field is if you have ordered a test and you need to interpret the test, and it may be difficult to interpret for each gene-drug combination how to adjust the dose. And that is the reason why we started a Dutch pharmacogenetics working group from the Royal Pharmacists Association in the Netherlands. In 2005, this group was started, and we had two aims. The first aim is to develop guidelines or recommendations, pharmacogenetic guidelines, based upon a systematic review of the literature. And it should give a recommendation on how to adjust the dose in case you have a test result if you are a poor metabolizer or an intermediate metabolizer for a certain drug. And the second aim was to integrate these guidelines in the electronic prescription systems and medication systems used by physicians and pharmacists in the Netherlands. Currently, we have 94 gene drug pairs summarized, made recommendations on this, and among this 49 guidelines, 47 appear to be actional interactions, meaning that you need a drug, an adjustment of the dose or a switch of the drug in case of being a certain carrier of a certain genetic variant. We have fully integrated these guidelines in the healthcare system in the Netherlands. Nationwide, it is included in the National Drug Database, which is used by all prescribing systems in the Netherlands and inpatient and outpatient care, and also by all pharmacies in the Netherlands. This is an example of a screenshot showing a prescription, electronic prescription for a patient for nortriptyline. And it appears that this is, which is an antidepressant drug, and this patient appears to be a poor metabolizer. And then the system gives an alert to the physician saying that you should adjust the dose and to decrease the dose to 40% of the normal dose based upon this Dutch guideline, a pharmacogenetic guideline. So we have guidelines available in the Netherlands. They are also made public. You can find them on the PharmaGKB website, and they are fully integrated in the healthcare system. Our colleagues from the US have also made so-called CPIC guidelines, a consortium of the Pharmacogenetic Implementation Consortium, and we have performed a recently we have performed a comparison of the guidelines, of the Dutch guidelines and the CPIC guidelines, and the conclusion was published in CPT recently, and it showed that the guidelines of the both group are similar, generally similar with regard to the therapeutic recommendation for most gene drug pairs. We are trying to further harmonize these guidelines with the two groups. So if you have a closer look to these guidelines and you separate them in preventing toxicity or improving efficacy, then it appears that by far the most gene drug pairs are related to the decrease of toxicity, so the decrease of side effects. There are some related to improvement of efficacy, but the majority of the gene drug interactions are related to preventing side effects. So one problem with this single gene test is that the population health impact is not only dependent on this association, but also dependent on the frequency of the adverse event and of the genetic variant. And that means that we need to take into account the number needed to genotype. And that is, how many patients do I have to screen, do I have to test to prevent one from having an adverse drug reaction? And for this 6-mercaptopurine TPMT example, we have calculated the number needed to genotype, and it appeared that for the hematological adverse drug reaction, leukopenia, the number needed to genotype is 200. That means that you need to test 200 patients in order to prevent one from having a leukopenic event. In TPMT carriers, variant carriers, this number, of course, is lower. The number needed to treat then is 5. And that, of course, has to do with the relative low frequency of the TPMT genetic variants, but also with the relative low risk reduction and frequency of this adverse event. So Dunnenberger recently showed that if you move from single gene testing to testing a pharmacogenetic panel, and you choose, let's say, a panel of genetic variants, then it appears that almost every patient, 95% of the patients, have at least one actionable genotype. So this approach seems to be much more efficient instead of testing single genes. And the price of genotyping went down dramatically in the recent years. So this has come into reach to test a panel instead of single genes. And with this in mind, we started an implementation study in the primary care, the so-called IP3 study. 200 patients were offered free genotyping. We choose a genetic panel with a series of pharmacogenes. We tested for 40 alleles. 40 pharmacies in the vicinity of Leiden were collaborating. And in fact, we found the same as Dunenberger. 90% of the patients at least carried one actionable genotype. And of course, this is also a very interesting population, but because about 10% of the patient carried 4 or more actionable genotypes. We are analyzing this data, but we already have shown that more than 85% of the recommendations from the Dutch guidelines were implemented, were accepted, and the doses of the drug were adjusted according to the guideline by the physician or the pharmacist. So with this result, we made an extrapolation to the Netherlands and to estimate the percentage of prescriptions what would need a dose adjustment if you would know the genotype. So we had we used national prescribing data from this organization in the Netherlands, and we calculated all actionable first prescriptions of patients, and it appeared that in the Netherlands about 200,000 first prescriptions needed a dose adjustment or a switch if you would have known the genotype of this patient. And that means that about 1 in every 19 first prescriptions need a dose adjustment, which is 5.6%. And in fact, when we showed this to our physicians, they were a little bit surprised because most physicians think that genetic variation, pharmacogenetic, is a very rare event occurring in, let's say, 1 every 1,000 patients or so. But I think this data clearly show that it's much more frequent than we expected. So until now, I told you examples of implementation of pharmacogenetic in primary care, but also at LUMC we have implemented pharmacogenetic testing. And I want to share with you an example on pre-therapeutic testing of DPYD in patients treated for their cancer with 5-fluorouracil or capecitabine. These drugs are frequently used in cancer, in a variety of cancer, colorectal cancer, head and neck cancer, breast cancer. It's an effective but toxic treatment. Grade 3 or higher severe toxicity is seen in 15 to 30% of the patients, and drug-induced lethality is 0.5 to 1%. The drugs, capecitabine is an oral drug, a prodrug of 5-FU. 5-FU is metabolized in the liver to inactive metabolized, and for this inactivation, the liver enzyme DPD, dihydropyrimidine dehydrogenase, is very important, and this enzyme is encoded by the DPYD gene. About 3% of the patients, 2 or 3% of the patients carry the STAR2A genetic variant, leading to exon skipping splice site variant, leading to exon skipping of exon 14, leading to a non-functional DPD enzyme. And these patients are unable to metabolize these 5-FU or capecitabine drugs effectively. So a pivotal study from Maarten Denen, published in the JCO, has shown that if you reduce the dose of these patients carrying these genetic variants with 50%, then the incidence of side effects is 28%, which is similar to patients not carrying the DPYD variant but receive a full dose. However, if you give patients, and these are historical controls, patients who carry the STAR2 genetic variant, and you give them a full dose, they have a very high incidence of toxicity. And this study was, the reason was so convincing for our oncologists that our oncologists at LUMC consider DPYD testing standard of care, and we have implemented a program where all patients receiving 5-FU or capecitabine should be tested pre-therapeutically before the treatment starts, and the physician alerts the pharmacist alerts the physician if the drugs are prescribed with no DPYD testing. So currently we not only test for the STAR2A variant, but we have expanded the panel. We are testing for 4 genetic variants, are using a gene activity score, and the dose of 5-FU capecitabine is decreased based upon the gene activity score. So it's decreased 25% up to 75%, depending on the score. So we have implemented this program routinely since April 2013, and we did a retrospective analysis of the first 300 patients, 18-month period. On this graph, you see the number of the percentage of patients being genotyped before the treatment was started, and we could show that in the end, about 90% of the patients were indeed tested before their therapy started. Interestingly, in the patients where the dose of 5-FU capecitabine was decreased, we observed no grade 3/4 toxicity in the patients with an initial dose reduction. However, some physicians are a little bit worried of undertreatment of patients, and then if they do not observe toxicity in the first cycle, they increase the dose in the subsequent cycle. But in the majority of the majority of those titrations, we observed grade 3/4 toxicity. So I think the dose titration upwards based upon toxicity is not a very good strategy. The DPYD screening was feasible, showed to be feasible in clinical practice, and currently is standard at our institute. We expanded this program to a national level in the Netherlands, started an upfront DPYD genotyping funded by the Dutch Cancer Institute and Alpe d'Huez. And the aim of this study was to determine safety, feasibility, and cost effectiveness of upfront genotyping. We already know that we can only predict 50% of toxicity using DPYD screening. That is the reason why we also exploratory started to study DPD phenotyping. 18 centers collaborated. The study started in April 2015. The goal of our first phase was to include 1,250 patients. The study phase 1 is closed. The first phase is currently closed, and we have included 1,160 patients. In the meantime, a new guideline from the medical oncologist in the Netherlands has been drafted. And surprisingly, this update incorporated DPYD genotyping. It was recommended to test DPYD before start of the treatment with the fluoropyrimidine. I can show you some results. These are not non-published results, so preliminary results from this study of the first 1,100 patients. 7.7% of these patients were DPYD carriers. And the grade 3 or higher toxicity was still 39% in carriers versus 23% in no carriers. The hazard ratio compared to historical control separated per genetic variant shows that there's a reduction from the risk of toxicity, grade severe toxicity, from 2.8, 2.9 to 1.3. However, for example, this one, we see that there's no reduction on toxicity, the risk of toxicity upon reduction of the dose. And we believe this is a 25% reduction of 5-FU capecitabine. We believe that the reduction of 25% is not, should be higher to prevent toxicity. As I said, many physicians are worried about undertreatment of the patient, and that is the reason why we also estimated drug exposure by measuring drug levels in the patient carrying DPYD genetic variants and patients who underwent dose decreasement of dose. And we can have shown in this slide that the drug exposure up on decrease of the dose was similar as compared to patients who were wild type and received the full dose. So currently at IUMC we are testing routinely for a variety of genetic variants. I already showed you DPYD, TPMT, but also in transplant patients are tested routinely. And about 2,000 patients per year are currently tested in our pharmacogenetics laboratory. We also started a genetic counseling and pharmacogenetics clinic at IUMC, where a clinical geneticist in combination with a clinical pharmacist and clinical pharmacologist are counseling patients and discussing their pharmacogenetic test results. Patients come with a variety of questions. For example, patient was a poor metabolizer, the patient is CYP2D6 like Bob Smith, and patients want to know for which drugs is it relevant. Can I take paracetamol safely? Should I inform my parents? Should I inform my children? This kind of questions are discussed with the patient at the outpatient clinic. I showed you the implementation program in Leiden. We have expanded this program to the European level, and we were very lucky to receive an Horizon 2020 grant from the European Union in order to implement preemptive pharmacogenetic testing in 7 European sites using the Dutch guidelines. And we will evaluate patient outcome and cost effectiveness in this study. This is a 50 million euro project. It started the 1st of January 2016 and has a duration of 5 years. The aim of this study is to reduce severe adverse drug reactions grade 3 or higher with 30%. We have formed a UPDX consortium with collaborating centers, collaborating scientists all over Europe. In this clinical study, we will include a total of 8,000 patients. That's the number we need to prove that we can reduce the sample size by the risk for adverse drug reaction by 30%. And the study is currently open. 7 centers are including patients. They are starting with pharmacogenetically guided prescribing, then switch after a period of 18 months to standard of care, or the other way around, in a way that every center can serve as its own control. We are measuring the incidence of adverse drug reactions and have introduced a so-called pharmacogenetic passport. Patients receive this passport, this card, the so-called safety code card. And the safety code card contains a QR code. You can scan the QR code, and the QR code contains let's say, the pharmacogenetic test results of a panel of genetic variants. When you scan the QR code, it brings you to the website of with the pharmacogenetic guidelines and gives you an interpretation how to adjust the dose for this particular genetic variant and this particular drug. So the current status of this project is shown on this slide. We started including by March 2017, and yesterday I checked the number. We are close to 3,000 patients being included in this study. I hope I have convinced you that pharmacogenetics is a powerful tool in prevention, especially in prevention of adverse drug reactions. And the implementation, its use in the clinic and in primary care, outpatient care, is feasible and has shown to be effective. And we hope that our European project will help clinicians to truly embrace the concept of personalized medicine. Thank you very much.